Okay. Hello and welcome to part 3B. We are going to be talking about antisense technology today. Okay, so what is antisense technology? All right, so we talked about the central dogma of biology last time, and we looked at how an oligonucleotide could block a messenger RNA. So literally this is how it happens. If you have a messenger RNA that's highlighted in green here, and if this messenger RNA encodes a protein that is not fit for fitness for that person, if it causes diseases, then what you can try to do is develop a conventional small molecule inhibitor, right? But we talked about some of those challenges because it's estimated only around 15% are druggable. So this is where antisense DNA comes into play. Because you know the sequence of the messenger RNA, you can therefore design an antisense DNA that would be complementary to that. You could synthesize that very easily on the synthesizer. It would form a duplex, right? A duplex in the cell. And then there is an enzyme called ribonuclease H that is ubiquitous. It's in all of our cells, also known as RNase H. And it's a very specific type of enzyme because it only recognizes RNA DNA duplexes. So it won't recognize DNA DNA duplexes. It won't recognize RNA RNA duplexes. Only recognizes RNA DNA duplexes. And what it does is it actually is a nuclease. So it actually will cut the RNA, which is exactly what we want, because if we cut the RNA, then translation cannot proceed. So RNase H only recognizes RNA DNA duplexes. Here is a structure of the RNase H protein, and you can see that it wraps itself nicely within the RNA DNA duplex, and there's a catalytic portion here which is mediated by ions highlighted in color there, which cuts the RNA. So antisense technology employs a synthetic oligo that's complementary to the mRNA, and one of the two main things can happen. So number one, we just saw RNase H cuts the RNA in RNA DNA duplexes. And number two, if you design an antisense DNA that still can bind to the RNA, but is not recognized by RNase H, that's okay too. You can do something called translational arrest, in which you get steric block of this duplex that prevents the ribosome from making proteins. Okay, so sounds fairly straightforward. As I mentioned before, this concept was, really developed in the 1970s. And here we are 50 years later, and we have challenges with using this as a potential therapeutic. So one of the main challenges with using oligonucleotides as a therapeutic, but especially for something like antisense, is number one is stability. So oligonucleotides are not the most stable structure. And this is because we do have a labile phosphorus-oxygen bond. We know this is labile. If we think of molecules like ATP, those oxygen-phosphorus bonds, those can be formed easily, but they can be broken easily. So there's an intrinsic issue with stability with oligonucleotides. The other issue, too, is that you have these negatively charged phosphodiester bonds. So if you have a long oligonucleotide, you now have a molecule that's polyanionic, and this is going to impact its cellular uptake across a negatively charged cellular membrane. So that's going to pose a challenge. Biodistribution. Suppose the molecule does get inside the cell, or if the molecule is circulating systemically within your body, it's unclear how the body will distribute that. And typically the liver will see these molecules as foreign and as toxic and will essentially start oxidizing and removing them from the body. In addition, we also have an immune system that is designed to keep foreign genetic material out. That's of course, that's a good thing. We've evolved to, to block out foreign genetic material. However, if we're trying to use oligonucleotides as a therapeutic, then our immune system doesn't know the difference. So the body can attack it inadvertently. And then finally, we can have something called off-target effects, where you actually target not only your area of interest within the messenger RNA, but you actually target perhaps similar genes that in similar sequence, or you target the wrong genes. So it's very difficult sometimes to achieve specificity and selectivity with oligonucleotides. So there's something called the Lipinski rule of 5. So you may have learned about this in some medicinal chemistry classes. If not, this was developed by chemists, Lipinski, who basically said that any type of drug that's developed has to follow these rules, which are multiples of 5. So number 1, the molecular weight of a compound should be less than 500 daltons. In addition, you don't want something that's too polar, so there should be no more than 5 hydrogen bond donors. There should also be no more than 10 hydrogen bond acceptors. And then finally, the octanol water partition coefficient, the log P, should be less than 5. So oligonucleotides have this issue because they're not small molecules, they're rather large. If you have an antisense oligonucleotide, this is going to be roughly 20 to 23 base pairs in length. This is going to be roughly 6,000 to 8,000 in molecular weight. And it clearly violates all of these rules that Lipinski made out. You have so many hydrogen bond donors and acceptors, and this thing is a very polar molecule. It doesn't really have a lot of hydrophobicity to it. So despite this challenge, chemists have been embarked with saying, well, can we look at a lot of these issues and try to solve them using chemical modifications? Can we design modifications that would function just as well or maybe better, but maybe, for example, fool the body into thinking that it's not a foreign genetic material? Or can we look at the backbone and remove the negative charge and add something that is perhaps neutral or positive so we can increase uptake? And can we look at different modifications that will increase the stability of the compound? So these are questions that nucleic acids are constantly asking ourselves to try to improve the properties. So I'm going to highlight a few examples of antisense oligonucleotides that have gone through these modifications. Okay, so this is on the left here, we have typical DNA or RNA, and one of the problems that we talked about is that you can have nucleases that come in, and nucleases promote the degradation, in this case, of RNA, or you can have water-mediated hydrolysis of the phosphate backbone, which is again mediated by nucleases. Okay, so stability is a major problem. So in the 1980s, there was something known as the first generation oligonucleotide, and this involved replacing one of the bridging oxygens with a sulfur. So you can see here we have a sulfur. And we call, in general, these groups here, not phosphodiester, but phosphorothioate. And the advantage of using a phosphorothioate was that you increase the stability of the oligonucleotide because it inhibits the nucleases. The nucleases, they can't recognize that sulfur. It changes the recognition within that active site. And the other advantage is that you don't really change the structure all that much. You still have your bases, they can still hydrogen bond, you still have your sugar. Some of the cons associated with phosphorothioates was the sulfur, the negative sulfur that's formed. So you can imagine you actually have a resonance structure between this oxygen, so you could have a different structure where you now have a nucleophilic sulfur. And this sulfur can actually cause some cytotoxic effects because it is fairly nucleophilic. So you can get non-specific binding within the cells, non-specific cytotoxicity. Another issue, and we'll talk a little more about this in the next course, is by introducing the sulfur here, you are now generating two different potential stereoisomers at this position. And introducing new stereoisomers at each phosphorothioate junction increases the number of individual molecules. So you actually lose specificity and you increase the likelihood that it could bind to a very similar target. So I'll just write diastereomers are formed. Okay, so this was in around the 1980s. So so war continued in the 80s, but in the 90s there were some very important modifications that were developed, and this involved sugar modifications at this 2' position, where an R group could be really whatever you wanted. And we looked at a couple examples in the last part, but very common ones involve using an electronegative substituent, so things like fluorine or an O-methyl group. What these would do is these would maintain the sugar conformation of an RNA, helping it bind really well to that molecule, but it increases the stability. So if you just sneak over here a little bit, you can see that this hydroxyl here with RNA actually can act as a nucleophile. But if you replace this R group, if you replace the hydroxyl group with an R group that has something like a fluorine or an O-methyl, that can no longer be a nucleophile. So the pros, again, you increase the stability and the binding affinity to the target. In addition, it's been shown too that if you add the fluorinated group to these molecules here, the immune system actually doesn't handle that as strongly. So it doesn't detect it as foreign genetic material, and it can therefore avoid the immune system, which is what you want if you want it to behave as a drug in the body. Some of the cons is that, like the first generation and native DNA and RNA, it still has trouble crossing the cell membrane. Okay, so some of the things that have been worked on since around probably 2010, a little earlier to present, involves saying, well, we still have this negative charge. Even if we make phosphorothioates, we still have a negative charge. Can we replace the backbone with molecules or entities that would be neutral or maybe even positively charged? Okay, and you've looked at a lot of different types of functional groups that meet these features and requirements. So for example, proteins use amide bonds. Amide bonds are stable and they are also neutral. Carbon-based linkers might also be a good option. And triazoles, those are neutral aromatic planar molecules that could be robust in replacing that linkage. So I work in this area too, in which I try to design different backbone modifications for our oligonucleotides. So definitely check out my research webpage if you have a chance. I'd be happy to chat with you also. So the pros are that you typically increase the stability of these molecules even further, because nucleases can no longer recognize a foreign backbone. So increase in stability, increases in binding, and sometimes you get increases in cell membrane permeability. Some of the cons that we have to be careful, though, is that if we drastically change the structure of the oligonucleotide, we want to make sure that's still bioactive. And the chemistry can be quite challenging. So it's not really there for prime time. There still needs to be optimizations done to a lot of these types of modifications before it might be introduced into an oligonucleotide as a therapeutic. Okay, so you can think of this like your, if you have a phone, right now I'm using my phone for this video, but every year there seems to be a new Apple or Android device, right? But you can see here that we start with first generation. It took 10 years to generate second generation, and now we're sort of entering that third generation of oligonucleotides, which involves not only replacing the backbone, but perhaps tagging it with small molecules. Whereas Apple, I think, is on their 11th or 12th generation, right? So a lot of work goes into this, and that just gives you a flavor of the different types of modifications. So let's take a look at one example here, which is called Morpholino. This one's pretty interesting because here's your regular phosphodiester DNA. What Morpholino is you have this unnatural heterocycle, this sugar. It's not even a natural sugar, oxygen and a Morpholino ring, and it's held together through this phosphate group that has a dimethylamine group attached. And under physiological pH, this would actually become protonated, and this would actually perhaps allow it to cross cellular membranes much better. So this type of modification is resistant to nucleases as well, and it's good at cell membrane permeability. It's not the easiest molecule to synthesize, however, and you really need to be an expert in designing these types of molecules. If you don't have the expertise in making it, you can definitely buy it, but it does cost quite a bit of money. So it's one of those situations where no more research is needed on it, but it's just costing too much money for researchers to really explore. Let's look at some approved oligonucleotides. All right, so the first one that was approved for an antisense is something called Vtrophene, approved in the late 90s. And this is an antisense oligonucleotide that targets a virus called the cytomegalovirus retinitis, which is a virus that targets the eye. Here is the structure of the oligonucleotide. I know it's quite hard to see, but what I just want to highlight to you is that we do have some modifications here, and these are phosphorothioates all throughout the molecule. And this virus is prominent in individuals that might have a weakened immune system. So patients who might have HIV or AIDS, or who have undergone perhaps something like a bone marrow transplant or chemotherapy, or perhaps have taken drugs that suppress immune systems, these patients would be the most susceptible to this type of virus. So what this does is this blocks the translation of key proteins in the eye that make this virus propagate. This was developed by a company that was used to be called Isis Pharmaceuticals. They renamed their company Ionis Pharmaceuticals in 2015 for obvious reasons, but they are the same company. The other thing I want to just mention is that you still have challenges with delivery, even with phosphorothioates, because you have a negatively charged molecule. So the way this is delivered is that it's actually injected into your eye with a needle. Okay, so it's not administered systemically. You cannot take an oral pill for this one. So the delivery method has been overcome by simple injection, but that certainly would not be pleasant if you think about that. A needle into the eye, who wants that? All right, so like 15 years passed, and then the next drug came on the market called Mypomersen, approved in 2013. This targets messenger RNA for apolipoprotein B, and it's a cholesterol drug reducing candidate. Apolipoprotein B is the primary protein involved in making low-density lipolipids. Or lipoproteins. This is also known as bad cholesterol. So if you ever get blood work done, you have to, typically you get your LDL and HDL analyzed, also developed by Isis and Ionis Pharmaceuticals in 2015. So I'm going to just pause this for a second. I think I should be okay. It's telling me that I may have hit capacity on my hard drive. Not sure. Let's see how this goes. Hopefully I don't get cut off. Mypomersen is another molecule that targets hypercholesterolemia. And what it does is that you have phosphorothioates, has modifications, but you also have different R groups on these sugars. So you're combining a first and a second generation oligonucleotide together. These different R groups equals to this OCH2CH2OCH3, which stands for this O-methoxy ethyl group, or MOE. And what this looks like is as follows. You have this O-methoxy ethyl group, methyl group, and it provides shielding, and it provides resistance to nucleases. So you have this water-mediated shielding. So you can think of it as a shield so nucleases can't come here, and it provides stability against nucleases. It also increases its stability to the target. And because you're adding 3 carbons, it's more hydrophobic. And you can get increases in cell membrane permeability. So that's another molecule that's on the market. This, we'll talk about the sugars a little later, but it adopts what is known as a C3' endo conformation. Okay, so just remember that name for now. We'll talk more about what C3' endo conformations are during the course pack. This molecule here called Ateplyrsen treats some forms of Duchenne muscular dystrophy, and it's a Morpholino type of molecule here. And the way it works is actually very different and novel from other types of antisense oligonucleotides. You may remember from biology that genetic information needs to be excised from exons and introns. Exons are the coding region, and introns are the genetic material that gets spliced out. So there happens to be a mutation in this genetic disease at one of the intron sites, and what happens is that the protein is not fully made because of that mutation. So using an antisense to target that mutation site allows for proper translation to occur, so it doesn't stop. And even though you have a mutation, you still retain around 50% of the normal activity of the protein. So this is much better compared to truncating that protein and getting no activity. Okay, and then the last one I'll talk about today is Forsen, developed in 2017. It targets bowel disease. It targets a protein called or a gene called intercellular adhesion molecule 1. It's involved in inflammatory disease. You can see the modifications that we're using are phosphorothioate, so we're seeing that a lot for sure. And we get enhanced stability because of that modification. So those are just highlights of some of the antisense oligonucleotides that are used as therapeutics. There's a lot, there are other ones too, and there are a lot more in clinical trials. So this is really an exciting time for oligonucleotide therapy. Okay, next session will be RNA interference and short interfering RNAs. See you then.